Okay, exciting times. Unit 6, Genetics, Keystone exam review packet. If you are in your reading packet, this is pages 7 through 32. Why doesn't it say what the workbook pages are? Well, it's workbook pages 3, 4, 5, 6, and 7. And then it's Study Island Modules 9, which is letter J, DNA and Genetics. And then Study Island Module 10, which is letter K, Heredity. This is also in your book in Chapter 6 and Chapter 7. So here's where we break away a little bit from being like Unit 6 is Chapter 6. We start to get a little bit further ahead in the book than we are with the units, but you guys are smart and you'll be able to keep up. The first question in the review packet here is describe how meiosis differs from mitosis. Use a drawing as necessary. In Chapter 5, we talked a lot about mitosis, which is making identical cells. Today, we're talking about meiosis, which is making your sex cells. A little hint that we heard Mrs. Rance, I believe, say was meiosex. Okay, you're making sex cells, which are sperm or eggs. So just as a review, mitosis, if we started off with 46 human chromosomes and then the cell went through mitosis, that could be something like a cheek cell if you bit your cheek, or a skin cell if you cut your finger, you would end up after the process of mitosis with 2 identical daughter cells, each with 46 chromosomes. These are all diploid cells, and when I think of diploid, I like to think that the number is double, sort of. All right. In meiosis, there's 2 stages of meiosis. It's called the reduction division. Meiosis is making sex cells. So we start off with a diploid cell with 46 chromosomes. We call this our 2N cell. It goes through meiosis 1 and is going to create 2 haploid cells, each with 23 chromosomes in there. I wrote 46 at first by accident, but it's 23 chromosomes in there, and we just say that that's N. So instead of 2N, it's N. These are haploid, and the way I remember haploid is I think of the word half. All right, so meiosis 2 then is going to go through those stages of meiosis again. It's going to go through prophase 2, anaphase 2, metaphase 2, telophase 2, and prophase 2, metaphase 2, anaphase 2, telophase 2, and you're going to end up with 4 genetically different haploid cells, and these are going to be either sperm or eggs, which are called sex cells or gametes, things like that. Now, it's just important to note that during meiosis 1, something called crossing over occurs. During crossing over, happens during prophase of meiosis 1, it increases genetic variation by exchanging chromosomal information. It's how you're not a clone of your siblings. If we look at these chromosomes here, you see you have a black one and a brown one. This is just an example. What is going to happen during crossing over is they're going to rub up on each other, and when they uncross, some of the black chromosomal material got kind of knocked off onto this one, brown chromosomal material rubbed on over to that one. So it exchanges genetic information. So at the end, when you get these 4 genetically different haploid cells, they have some variation. It's how your eggs get scrambled a little bit, the sperm gets the chromosomes a little bit differentiated, so that way you're not an exact clone of your brother and sister. Number 2. Think about number 2, and it kind of just helps us review some of the essential vocab that we went over in chapters or in Unit 6. Create a Punnett square to demonstrate the potential offspring of 2 parents who are heterozygous for brown hair. So remember, heterozygous means other, so that means we're going to have a big B and a little b and a big B and a little b. If we do a Punnett square with these guys, we're going to have a 1 to 2 to 1 ratio. That's what we always get with a heterozygous cross. Big B, big B, big B, little b, big B, little b, and then little b, little b. Big B is homozygous dominant genotype. That's its genetic makeup. And its phenotype is that the offspring is going to have brown hair. Big B, little b is a heterozygous dominant genotype. the phenotype will also be brown hair. And then little b, homozygous recessive genotype, and the phenotype would be blonde. So your phenotype is your physical characteristics, your genotype is your genetic makeup, and even if you have two brown-haired parents, if they're heterozygous for their brown hair, you have a 25% chance of having an offspring who has the two recessive alleles. That one comes out with blonde hair anyway. So that's just a quick Punnett square review. If you're confused about Punnett squares, take some time to go back to the Unit 6 resources and just review a little bit of that information, because there definitely will be some Punnett square questions on the Keystone. And as always, make sure you're using your scrap paper for the Keystone. All right, describe Mendel's 3 basic principles of genetics. We already kind of did with the law of dominance. When an organism has 2 or more alleles for a trait, the allele that is expressed in the phenotype is the dominant allele. So in this example, big R is red, big R is red, little r would be white. In the law of segregation, different alleles for the same trait separate when gametes are formed. So if mom's genotype is big A, little a, when her gametes or sex cells or eggs are formed, these are going to separate. They're going to segregate from each other, separately from each other. So one egg will have a big A, one egg will have a little a. And then what I think is the most kind of confusing, because it's a little bit repetitive here, the law of independent assortment states that when pairs of alleles separate, they do so independently. So if mom's hair is big B, little b, and mom's eyes are big G, little g, they're going to separate independently from each other, and you could end up with any combination of those alleles.